say it's good enough.
It's good enough?
Yeah.
Okay. If it's good enough, then we can start.
Yeah. Today, Azat will give us a talk about constructive natural evolution. Azat's thesis is about the philosophy of biology, especially about the evolvability, and this topic is also talk about the developmental biology in contrast with neuroscience, natural selection. Yeah, let's start.
Okay, so thank you very much. Maybe we can close the door, please. Yeah, thank you. So again, I'm Azat. I'm a PhD student here. Maybe it's more for YouTube than for you. So my talk is on constructive neutral evolution, and this is mostly what I have been doing during my master's, for my master's dissertation. That's why I put the logo of Université de Bordeaux and Philosophy in Biology and Medicine team at the university where I did my master's. And as Wen rightly said, it will be, in a sense, on this broad topic of developmental constraints, some sort of internal properties which impact the evolution in contrast to natural selection, but in very broad sense, and it won't be connected that directly to what people usually write in Evo-Devo, while I think these are all quite related topics. So first, constructive neutral evolution. What the hell is that? It's a bit complicated because it's a very long name, but in fact, it's a specific theory which was developed in the 90s by a group of biologists, and the term was coined by Arlen Stoltzfus in 1999. And this is a theory which aims to explain a non-adaptive, hence neutral, origin for novel functions. Novel functions, that's why it's a constructive neutral evolution. And functions here are understood as parts or processes which contribute operatively to the fitness of the organism. So the focus here will be mostly on causal role conception of function rather than selected effects. And one of the ways to read all this constructive neutral evolution is as a specific case where the causal role function diverges quite significantly from selected effects function. And to explain what is this specific theory, I invented a toy scenario, which I think helps to kind of grasp what's it all about. So imagine you have a nice house with an attic, and the attic is fully functional because you use stairs, which are inside the house, to reach the attic. And it's all fine, it all works, that's it. And then one day, you put a ladder in front of your house. Maybe because you decided to repaint your house, now you need a ladder to repaint your house. Maybe you just have this ladder and it's so big, so you have no space in your house to put it anywhere, so just put this ladder on your house. And it doesn't change anything. Maybe it helps you, I don't know, repaint the walls, or just doesn't bother you, but it doesn't add anything to your usage of attic and to your capacity of reaching the attic. But then imagine one day, the stairs which are inside your house, they are broken, what is shown on the picture on the right. And what happens in this case? In this situation, you will still be able to reach the attic because you have a ladder outside, right? So from the point of view of how well you can use your attic, nothing changes. You started with the situation where you were using your attic, and you ended up with a situation where you are still using your attic. The only difference is now you have a ladder in your house, which became essentially functional for the house, because without this ladder outside of the house, you cannot reach the attic. So in a sense, the system of your house, it's a bit complexified. Now it has new components. And if we imagine an evolutionary biologist who reads the situation in the very end and just sees a house with an attic, broken stairs inside, and a ladder outside, he may very well say something like, oh, of course, there was a house with an attic, but then stairs broke, and in order to be able to use attic, this person brought in ladder. So ladder was, in a sense, adaptive, because now you see it has an essential function of allowing you reaching attic. But this is not what happens in fact. In fact, you first just sort of happily and contingently got this ladder, and it happened so that it allows you to perform some functions and to prevent the negative effect which would have been associated with the stairs broken down. But because it just happens a way, it may also happen this way that stairs will indeed break down, and if so, then the ladder will become an essential component of the house. And this is the general idea. And later, I will present biological examples which follows the same scenario and show why it is, how it may affect our ideas about how organisms evolve new functions, new structures, new properties, etc., etc., etc. So now a brief plan for my talk. First, I will try to sketch the way biologists speak about the evolution of complexity, and I will introduce the notion of so-called unnecessary complexity, as it's sometimes called, especially in the context of eukaryotes compared to prokaryotes. Then I will switch back to the constructive neutral evolution, or CNE, as it's usually called in literature. I will present a couple of examples, and I will also argue that in fact it has two different subtypes depending on the specific scenario of what's going on. Then I will talk about the connection between the constructive neutral evolution on one hand and the robustness of biological system on the other hand. I will explain what it means, and maybe I will also talk a little bit about evolvability. And if you see anything gray shaded, usually it means that it's rather secondary, or in this plan it's something which is not developed well, so I will not present it fully. And then I also thought quite a lot, and that was one of the things present in my master dissertation, is how we should argue in favor of an argument by constructive neutral evolution rather than an argument like more traditional argument by natural selection. And this is not what I'm going to talk today, because I think I just don't have enough time. And in the end, I'll try to somehow sum up everything and maybe put some questions on, because it's a work in progress, so I don't know where to develop this project and what to add and what maybe to remove. So, okay. What is this unnecessary complexity, and how in general biologists think about complexity? Well, evolutionary biologists started thinking about complex systems, complex structures, etc. It's been quite a while. The most well-known, the most canonic examples comes straight from Darwin, where he discusses the evolution of something really complex, for example, mammal eye. If you look at the mammal eye, you see that it has a lot of elements, everything is connected with every other thing. They're so tightly correlated and they have so many interactions that it puts a major problem on how such a complex thing may have evolved. And one of the things why this is interesting is because this complex thing, very complex structure, the eye, it also performs a very complex function. It does a lot of incredible stuff. It can, I don't know, you can look in many different directions. You have high resolution vision, which is adaptive for the, I don't know, general amount of the light, which you see. So you can, I don't know, use your eyes in a dark room, in a bright room, on the sun, maybe underwater, whatsoever. Still, the eye shows you the high resolution picture of what's going on outside. It's a very complex function. And indeed, there is a very complex structure, and the way to tackle this problem, it was suggested by Darwin, and it was indeed found that probably eye evolved in this fashion. So what's the idea? The idea is that during the course of evolution, the evolution of an organ, for example, it starts with a simple organ, which does simple function. For example, we just had a screening pigment, just light detection pigment, which was directing light from any direction. Then the next step, screening pigment appeared, which allowed to detect light only in one direction. So new element, complexification of structure, comes because it allows performing a more complex function, not just detecting light anywhere, but in a specific direction. And then we can see that by several steps, indeed, structure complexified, but the function itself also got much, much more complex and refined. This is why we observe such a complex structure as a mammal eye or insect eye or any other eye, because during the course of evolution, consequently, many, many different steps led to the performance of complex functions, which are adaptive, because if you have an organ which can perform complex functions, you are more apt for example, changing environments, etc., 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 etc. So this is the traditional topic which interested evolutionary biologists, but in fact, not all complex traits are like this. sometimes we see something very different. And the best examples come from the comparative genomics of eukaryotes and prokaryotes. So let's see what's going on over there. If we just take a prokaryotic genome and try to look what's inside, and then look at the eukaryotic genome and just look what's inside, we'll see something very odd. So what's on this picture? On this picture, you can see just a comparison of the genome maps of 4 organisms, E. coli, so single-cell prokaryote, baking yeast, so single-cell eukaryote, and then 2 multicellular organisms, this is fruit fly, and humans. And they are all centered around this red box, which represents an RNA polymerase II gene, which is a gene which is shared homologically by all these organisms. And all the green boxes, they represent different genes. And if you look at the genome of E. coli, you will see something which looks reasonable. You just see a lot of genes one after another. That's it. Sometimes there is some space between them, but nothing interesting. If you look at baking yeast, you see something very much similar. You also see a lot of genes, right one after another, but sometimes you see strange stuff. These are small blue boxes, which are called introns, and this is the thing which you observe a lot in eukaryotes. It means that inside a gene, which codes a protein, there is a small region which doesn't code for the protein, so it's just inserted, and it has to be removed on the later stages of gene expression. When after the gene is transcribed, it has to be removed before the protein gets synthesized, because if you leave intron, usually you get a non-functional protein. So it's important, and you see that sometimes you see those blue boxes, but not a lot of them. But if you look at fruit fly genome or human genome, you see that almost every gene is just full with all those introns. And obviously, if you compare multicellular eukaryotic genome on one hand and single-cellular prokaryotic genome on the other hand, you see that the eukaryotic genome is much more complex because it has this thing. It has introns which has to be spliced out. And it's not clear why, because it seems as just a sort of unnecessary addition, because why couldn't you just put functional genes inside it and just do it like it's done in prokaryotes? Why there are those inserted fragments which just seem to complexify the situation without giving any, at least obvious, adaptive benefit? And obviously, if you look at the gene expression sort of biochemical pathway, you will see that indeed, if we are talking about eukaryotes, which have introns, they have an additional step of removing those introns, which are yellow on this picture, in order to get a functional nature matrix RNA, which will code for the protein. So in the end, we will have a functional protein, which is represented as this colorful blob in this picture. So you need an additional step in this gene expression process, and it's also not clear why. And if you look how this splicing process is done, you will observe that this is something very, very complicated. Here is just to scare you off, because it's just a scheme, quite simplified, of what's going on when a single intron gets spliced out. You see that there is a cycle. So on the left, you have intron inside RNA, and on the right, the intron is spliced out. And you see that there's interaction of a lot of different particles in a very complicated manner, which have to form a lot of complexes in order just to get an intron spliced out. And on the right, you see that these particles, U1, U2, U3, and U4, and U6, they all have not a single protein, but an RNA molecule and a lot of different proteins. So every name here is a different protein. So you see it's a huge complex, possibly one of the largest RNA protein sort of molecular machines in our cells, which does just this job of removing the intron. And to remove every intron, you have to assemble this whole machine just to do this quite simple stuff. And what's even more shocking is that those introns sequences, they are sometimes found in bacteria. They're just very, very rare. And if you look what happens in bacteria, you will see that they don't need any proteins. They don't need any machines, the introns just splice out themselves. So the whole reaction is catalyzed by themselves. So you see that in prokaryotes, this reaction is catalyzed by the intron itself. And in eukaryotes, you need to assemble this machine consisting of hundreds of proteins, many RNAs, consume a lot of energy because you need sometimes to spend ATPs and JTPs just to get this thing running. And it's not so clear why is this going on, why we have such a complex thing on the left and such a simple thing on the right. And the way to represent this is maybe to compare the complexity of behavior we observe and complexity of mechanism we observe. And we will see that if we look on something like I and the evolution of I, we would start with a simple behavior and simple mechanism, and we will go towards complex behavior and complex mechanism. But if we look at something like spliceosome, this machine which was designed to splice out introns, we'll observe that it's something weird because it seems that behavior doesn't change dramatically, just splicing out all the introns, but the mechanism gets much more complex. So the standard sort of Darwinian decomposition of complex function into many simple functions which add one after another just doesn't work because we don't have complex functions. It's just some sort of unnecessary complexity. And what to do with it? And many different answers were proposed to these kind of situations, and in particular regarding this complexity of splicing. First answer, which may be the simplest one, is that maybe we think that this splicing is not so complex in, like the function is not so complex in eukaryotes, but in fact it is. Maybe all those hundreds of proteins, they perform some functions, we just don't know them. And this is why it's so complex. In fact, it's all adaptive. Maybe they regulate this mechanism, which is actually true because if you look, it's very well regulated and there is a lot of regulation pathways. Maybe they allow something what is impossible in prokaryotes, and this is also true. For example, in eukaryotes, because the process is regulated, you may sometimes splice the protein differently, ending up with different protein structures starting from the same gene, which gives you some sort of functional diversity and diversity of proteins, which is not observed in prokaryotes. This is true, but it's still, it seems that it's sort of the answer is not given. It's just saying maybe there is some function we don't know, but unless we show the function for every protein, for every complexification, this answer will not be satisfactory. Second idea is that maybe the fact that mechanism is complex, maybe it has some sort of more systemic beneficial traits. For example, maybe the fact that this intron is spliced out by hundreds of proteins makes it very robust. Maybe because there is this machinery, it will splice it out in any environmental conditions. It will splice it out maybe even if there will be some mutations in this intron or some other proteins which may be beneficial for the organism, which is also a valid hypothesis. But also it may be that this complexity just evolved neutrally. It just happens so. Maybe there is a trend for these protein or RNA molecular machines to accumulate many different subunits. And it just happens that they grow in course of evolution. But then the question is, what are those strands and where do these strands come from? Why do they exist? And in this talk, I will focus on this third answer. And because I think precisely constructive neutral evolution is one instance of these sort of evolutionary explanations in order to explain complexity. And first, I'll make a very brief literature review on already quite well-established theories regarding how complexity may evolve in a non-adaptive way. And first is the most general answer given by S.J. Gould in his book, I guess, Full House, where he has his Drunkard's Walk theory of complexity, which says that if we look at a large-scale evolution, starting from the first cells leading to the contemporary times, we expect that we will start with very simple organisms, because it's very unreasonable that we will somehow magically start with complex organisms. But then, even if the evolution of complexity is a purely stochastic process, we will observe that starting from this rather sharp situation where all the organisms are very simple, during the course of time, because it's purely stochastic, but it has its sort of one left wall, that there is some minimal complexity for a living organism, then we will expect that this distribution will just move to the right, and we will observe more and more of more complex organisms, and that's it. And it's a very interesting answer, because it presupposes almost nothing about living organism, but it's also, in a sense, not satisfying, because it doesn't explain how precisely the complexity evolves. It just says that it's not unexpected, that it will increase during the time. But it doesn't say anything about the precise mechanism. And the more specific thing is the idea by Daniel McShee and Bob Brandon. It's their so-called zero-force evolutionary law, which is expressed in their book Biology's First Law, where they say that in evolution there is sort of general trend towards increasing variety and diversity of parts inside an organism and organism in a population, or maybe I shouldn't have written a population, just organisms in the outer world. And it's also very expected that if you start with an organism which has, I don't know, 100 parts and they are all the same, then during the course of evolution, it's expected that some variations will accumulate and you will have more part types. So the diversity of system parts will increase. At least there will be some sort of just general trend coming from the fact that all the replication process are stochastic towards the increase of diversity and variety. But it's also, it's true in a sense, but it's very minimal. It only talks about the diversity, and in their account, the complexity is sort of reduced to just a number of part types in a system. It doesn't say a lot about whether there are some functional interactions or some hierarchical structures. We don't know anything about this. So it's, in a sense, it's good, but it's not what's going on, at least not everything, what's going on in constructive neutral evolution, at least it seems to me. And this is why I moved to the second part. And feel free to ask any questions, especially if there's something about biology which wasn't clear, because unfortunately I think it's quite a technical talk, as you may have remarked. And I want to be sure that you understand, at least generally, what's going on. So what is constructive neutral evolution? I already gave you this general idea about attic and ladders, and the fact that you end up with having something which is very essential for your functioning. It contributes to your fitness, but there was never a single moment in the evolution history where this thing was selected for doing this particular job. And I will present 2 or maybe 3 examples, which I think you should know about. The first one is the evolution of gene duplicates by their sub-functionalization. What is it? During the course of evolution, genes sometimes duplicate, which means that there is just a second copy of a gene which appears somewhere else in the genome. And interestingly, if we look at the statistics of how this process is going on, we see that these duplicates, they have a tendency, especially in multicellular eukaryotes, to accumulate. We observe that there are a lot of them which just accumulate in the genome and do something. And it's not clear why this is going on, because we would expect that sometimes duplicates are accumulated, but sometimes they are lost, because by a mutation, a duplicate may just get lost. And the observed numbers indicate that there are much more genes which are duplicated, which are accumulated, than we would expect if there was just this equilibrium between duplication mutation and the loss mutation. And there are 2 explanations for this process. First is the so-called neo-functionalization, which is the idea that maybe when there are 2 copies of the same gene, Now, because the selection pressure is sort of not working on both genes, or not on a single gene, one copy of the gene may evolve some new function and do something else, which may turn out to be beneficial for the organism. And now, because there are two genes, even if by this new function it loses some of its past functions, it's not bad because we have a second copy. So in a sense, maybe this duplicates, they just sort of increase evolvability of the organism. And this may be the case, and in the course of evolutionary history, we have observed these kind of events. But still, if you look at the statistics, it's not enough, because adaptive beneficial mutations, they are rare, much more rare than loss mutations. So it's still not explaining why there are so many of them. And the final answer, which it seems to be sort of consensus amongst population biologists, that at least it's important and it contributes to these process of evolution by gene duplication, is that it's called subfunctionalization. The idea is that most of the genes, they don't have one simple specific function. Very often they have sort of complex function. For example, a gene, a transcription factor, it may be expressed in your brain, in your gut, and in your skin. And probably if it expresses in this way, it means that it has three regulatory regions, one for your gut, one for your brain, and one for your skin. And if it allows us to see this gene as performing a complex function, which consists of three regions which should be functional for this gene to be expressed in all parts of your body. And if this is the case, then if you have two copies, maybe one gene will lose the first function and it will stop, it will lose its expression in the brain. But because there is a second gene, it's not bad. But maybe the second gene will lose its second function. So we'll stop expression in guts. And again, it's not bad because you have the first gene. So if it goes by this pattern, you observe that in the end, you end up with two genes, and both of them become essential. Because if you lose the first one, you no longer have a working gene in your guts, which is bad. If you lose the second one, you no longer have a working gene in your brain, which is also bad. So you started with one gene, which sort of contingently duplicated without giving any advantage to you. And then you ended up with, because they sort of deteriorated and sort of the complex functioning got divided into two subfunctions, you ended up with two genes, both of them essential for your functioning. So we see there is a complexification, there's an addition of a new element which never was adaptive, in a sense. That's the first example. The second example is precisely about this formation of these insane complexes such as spliceosome. And the idea is that maybe if we know that many proteins interact with each other in our cells, it just happens. And sometimes we know that two proteins start lumping together just for no reason, because it just happens, because proteins interact with each other. And it's represented on the right. So there is a sort of first step of just promiscuous binding of two proteins, and it may happen. But if you have this kind of binding, because there is binding, their free energy is lower, it means that maybe there are some mutations which would destabilize single proteins, but they would not destabilize a protein complex. Which means these mutations, which initially were deleterious for proteins which don't bind, now become neutral when there is this protein complex. So if you have this kind of situation, and you have this binding, you would expect that these now neutral mutations will start accumulating by genetic drift. And you will end up with a protein complex which needs binding for its functioning, because without binding, this mutation just will destabilize the protein and deactivate it. So you see again the same idea. You started with one protein doing one function, and then just because there is this promiscuous binding of many different proteins plus the tendency to accumulate neutral mutations, you end up with the two proteins forming a complex which is now essential for performing this function. And this may be the explanation for this thing such as spliceosome. This, I mean, it's a bit of hand waving because as far as I know, we don't have a final answer on spliceosome, but we know that something like this happened for some proteins. Because for some proteins, we were able to go through to reconstruct their evolutionary history and indeed find that there was first event of binding and then the mutations which would have deactivated the protein accumulated. Third example, I think I want to talk about. And okay, so I presented two examples, and it seems to me that they are of two different subtypes of sort of how did this happen. In the first case, with the duplicate genes, there was just direct redundancy, which allowed this process. Because you have two copies, two identical copies, this is redundant. So you may lose some functions of any of these copies without any negative effect. But in the second case, it was something rather different. It wasn't a creation of redundancy. It was that just this sticking of a new subunit, it somehow buffered the mutations in the other subunit. So it was some sort of interaction, buffering interaction. And this is just, it seems to me, two ways how this process may go on. And I haven't found any third way to do so. Maybe there exists one, but it seems to me that at least in the examples which we know, we observe these two ways. Yeah. And then with these examples, we can make sort of a conceptual structure of how CNE explanation works. And here I'm drawing quite heavily on the articles by Tyler Brunet and on the original formulation by Stoltzfus in '99. So how it works in general. Again, this is something I already said many times, but now in the most possible general terms. So CNE presents sort of complexity ratchet, which generates new dependencies between different components of the system. First step is that one component, A, is able, sort of promiscuously, to neutralize the deleterious effect of some possible mutations in B. And in genetical terms, A may be called a pre-suppressor because it has capacity to suppress the effect of mutations in B. But it does this before these mutations ever rise. That's why pre-suppressor. And here suppressor doesn't mean suppressing in physiological sense of reducing activity, something like this. It's just cancelling of the effect because it's a genetical term. Second, if we already had this situation, we expect that neutral mutations are going to accumulate. And we observe that mutations in B are getting fixed by drift. And every mutation makes B's functioning more dependent on A for its own functioning because more of these mutations which would have been deleterious, mainly mildly deleterious in B alone, now get neutral, so they get fixed because they're neutral. And after we have accumulated a significant number of mutations, A is now an essential contributor to the function of B, and if B has an important function in the organism, then A is now under strong purifying selection for this pre-suppressing neutralizing capacity towards B. So we end up with these situations where there is an entity which is under strong purifying selection to do some task, but it never evolved to do this task in some sort of positive way. So that's a general explanation. And here I think this is one of the places where I kind of stopped. And now I don't know what to do further. So I'm thinking of what to do further. And one way which I want to talk about in this talk is to try to look more closely on the specific cases of CNE and to see how to make like this sort of line-by-line comparison between adaptive explanation and CNE explanation to see why we prefer CNE or adaptive explanation or vice versa. Because for me it seems important because it is something about the fact how we infer at all that some structure is adaptive or not. And the second idea is that it seems to me that this process, constructive neutral evolution, is very much related with what's called robustness of biological systems. And let me explain how. So, what is robustness? Robustness is usually defined as systems' ability to properly function despite some external or internal perturbations, right? And it's maybe defined in regards to any sort of perturbations which may be observed. And one of the types of robustness is genetic robustness, which means a lesser sensitivity to the effect of deleterious mutations. And surprisingly, this is one of the steps of the CNE, yeah, right? These first step of CNE is that the system has to be sort of genetically robust, but with the difference that when we talk about CNE, you're interested in two components, one sort of which makes component A, which makes B more robust towards mutations. And this is straightforward similarity, but also robustness may be defined to something else. For example, it may be defined towards environmental perturbations. And there is some literature on the way how these different types of robustness relate to each other, because it seems that very often, if there is a mechanism which makes something more robust towards mutation, will also make it more robust towards environment and vice versa. And I think this is interesting because if it is indeed very often the same thing, that means that selection for environmental robustness will also lead towards more genetic robustness. And if it will lead towards more genetic robustness, it will lead towards CNE, which will mean that...
which will mean that CNE is not neutral.
Yes, this which means that it's not neutral, and which raises this problem that it's not neutral in which sense? Because if we say that there is still one sense how to say why it's neutral, because it was this robustness mechanism, it was never selected to sort of suppress these specific mutations which did accumulate. Because this is one way how people sometimes argue in favor of some robust systems. They say, look, our organisms, they are under constant mutational pressure, which means that they accumulate or they have deleterious mutations or mildly deleterious mutations, which are not selected against by natural selection, for example, because the population size is quite small. And in this case, maybe if you already in this situation of accumulating a lot of mildly deleterious mutations, you have a new mechanism which will allow you to neutralize the effect of all these mildly deleterious mutations. And in this case, robustness will be selected for. And it, in a sense, won't be CNE because it won't be neutral. But I don't see if there is a border between these two processes, and whether we should put a border between these two processes, and if we should, how. And this leads to one of the open problems I have now, is that how we should distinguish these two phenomena, selection for this sort of robustness mechanism which should protect us from deleterious mutations, and sort of pure CNE, or maybe both of them are never pure in the world, and we always have some sort of mixture. But again, maybe is it possible to somehow make a population genetic model of this process to show that they are expected to show different dynamics, or maybe they are showing the same dynamics? Then it's even worse, because it means that there is no way to distinguish them unless tracing very specific evolutionary history. So this is one of the problems I have right now in this project, what to do with this thing. And yeah, and second is that, yeah, it's also interesting because this robustness as talk, it's usually done by people in systems biology, whatever that means. And usually they say that this is something which is indeed favored by evolution in many places and in many systems. And they even claim, as Kitano does in one of his seminal papers on systems biology, where he was one of the first ones to really promote this label, that this is some sort of fundamental design principle, which is sort of necessary for any living being just to function in a constantly changing environment. And in this situation, robustness is regarded as something beneficial, something which is favored by selection. But CNE is a sort of, it has the opposite aim. It's aimed to show that this robustness, in fact, can evolve at least to a degree neutrally, and then just get maintained by purifying selection, even if it just evolved neutrally. And it seems to me this can be a sort of research program for evolutionary biology. I don't know whether it makes a lot of sense, but to me, it seems that it's an interesting question to sort of see to which degree it evolves neutrally and to which degree it's under selection and whether this matters or not. And yeah, this is one thing. And the second thing I want to talk about is that it was also, there is work by Andreas Wagner in Zurich, who claims that robustness may promote the genetic variability of the population, because if you have a robust phenotype, you may accumulate in population, not inside one organism, but in a population, you will have more genetic diversity because you can explore many more neutral mutations in a population. And if you have this kind of situation, then you increase the chances of the population to evolve some sort of evolutionary innovation, some sort of new adaptive function, something like this, because you're just exploring much more genotypes when you have a robust phenotype. And it's interesting because it links robustness to the evolvability of a population or species. Evolvability understood as sort of disposition to evolve some things rather than the other ones, and it seems to increase one. But here again, robustness is just taken as given, and it's studied how does already given robustness lead to increase or decrease or some change in evolvability. But again, CNE is sort of studying the previous step. It's interested how this robustness ever appeared, because it seems that it's sort of an argument which says that we will expect to observe robustness even if it's never beneficial in the strict sense of being selected for. And yeah, and I think, yes, this is the picture from a paper by Brunet and Little. They also hint towards the fact that these two explanations, they seem to have the same elements, but in different order. So the more traditional explanation on top, which they call error-correcting evolution, will be something like this. There is a mutation which is deleterious, which creates a selective pressure for evolving sort of suppression mechanism, sort of buffering mechanism, which will cancel the deleterious effect of this mutation. And CNE claims that it's actually the other way around. That first you just contingently acquire some buffering capacities, and then this allows you to get fixed some mutations. And you end up in a sort of same situation. And as I tried to show, it seems to me that these two processes may go on the same time. So it's not only just that they sort of, you can trace different stages, when first it's error correcting, then it's constructive neutral evolution, or vice versa, but that it's, at least for me, I don't see whether there is really any difference between these two, because it seems that if you have the first one, you immediately have all the conditions for the second one, and vice versa. And I think that goes to the conclusions, where, so I try to analyze the sort of conceptual structure of CNE and claim that there are two subtypes, which it makes out by accumulation of some redundant structures or by evolution of some sort of buffering mechanisms. CNE explains the non-adaptive evolution of genetic robustness, but it also may originate from this compensatory mechanism for the accumulated deleterious mutations, which questions all this distinction between error-correcting evolution and constructive neutral evolution. And it leaves us with some open questions. The first one I already stated, how to distinguish, should we distinguish CNE from the selection for robustness? Second, whether some mathematical modeling may help us or not, and if so, how it should be done, and what should we model, what questions should we ask? And the last one is just the question sort of about evolution as it happened. How important is this phenomenon? How general it is? How important is its contribution? Because there are many examples, not only this gene duplication and protein accumulation, there are also examples on higher levels. For example, it was proposed again by Tyler Brunet that some sort of maybe even organs or tissues may have evolved in this manner, or even ecosystems, and maybe some of these symbiotic relationships between the organism evolved by the same manner, because some interaction was just occasionally happened, and then it turned out to be essential. And yeah, I think that's it. So thank you for your attention.
Break, okay.
And I wanted to thank all my collaborators from Bordeaux for giving a lot of very helpful input, and Fridolin Kos for supervising my work as a master student.
Can I do a pause or end the stream? I don't want to end the stream.
I think you can turn off.
Oh,
Any questions? Yeah.
Oh, no, not cool. No, good.
Thank you. Thank you so much for your talk. Super interesting and give me the possibility to think about a lot of stuff that very interesting for me. And first question is very a curiosity. What do you think? I don't know if you notice term expectation that is called.
Acceptation.
Yeah.
Yeah.
And it's if I remember correctly, it's something that you have a character today that increase the fitness of an organism, but at the beginning was not selected for that kind of fitness.
Mm-hmm.
Okay, I think it's something very different from what you say in the constructive neutral evolution, because here. No, and I would like to know if you, if it's interesting to make a difference between the two, because I think, but this is an impression, we have different kind of stochasticity or nonetheless the contingency is in different places concerning this expectation by good and this idea of the constructive neutral evolution. What do you think? It could be interesting to compare and to see the differences.
Yes.
And,
In fact, there is an article about that. It's not all about that, but one of the sections is about that. It's again by Taleb René, where he explicitly compares constructive neutral evolution and this idea of acceptation.
okay.
And he distinguishes, he says that in this acceptation by Gould and Verba, I don't remember, there is actually two things which may be. First is that there is something which was selected for one thing and then it got used for something else.
Yes.
And the classic examples of a bird which uses its wings to shadow the water and then to see fish when there is sunlight. This is something like this, because wing was selected to be big because you need big wings to fly, but now you can use it to shadow the water to see fish and hunt fish. But there can be something else when something appears neutrally and then started being used.
This is
Yeah, it starts being under negative selection, and in the end starts being something which contributes to the fitness. And this is, it seems to me that CNE may be this kind of thing. So in a sense, this kind of acceptation will be more general term, and CNE will be one of its subtypes. But it also seems to me that there is, it's more specific because it talks about this accumulation of parts or processes or something like this. And it's also more specific because it has this kind of ratchet that you start, and then it explains how you can, starting from small dependency, evolve to a very big dependency, which is in itself is not contained in this idea of acceptation, it seems to me.
Yes. Thank you. We can move to that.
Is that done?
Yeah, it's a following, because if I understand correctly, the point about acceptation is that you cannot understand them as some select for robustness. No, you don't. They are they are useless before. But in the first case, for example, you can say, okay, if I don't say
First case is
gene duplication.
yeah.
If I don't stay at the level of the gene, if I stay at the level of the gene, the function of the gene is not selected. So from the unit of selection of the gene, I agree, not selected. But from the unit selection of the genome,
Yeah.
I could say I select for redundancy because robustness. So
Yeah.
at least for this case, I'm not saying for all case, but that's interesting because if it's a question of level of selection, you can do a mathematical model and look if it's explaining this it's a multi-level selection, at least for that kind of case. But maybe it's not the case for all kind of CNE, though. But this case, you know, I was waiting all your presentation. When will you say you select for robustness? But of course, you stay at the gene. You said the function of the gene is not selected, which is true. But at a higher level, it could be selected.
Yeah, but it also seems to me that in a specific, when you talk about just gene duplication of two specific genes, it's why people quite often agree that subfunctionalization actually occurs, is that because when you just have not a whole genome duplication, but just duplication of one single gene, the mutational pressure will be actually very, very small, because it will take quite a lot of time even to accumulate neutral mutations. And if the mutational pressure is small, because the gene is quite small, then the general selective pressure will be also very, very small. And then it's hard to argue that it was selected for robustness, because there was no actual mutational pressure, because it was only on one small region.
Yeah.
While if it's something like, for example, a protein binding, and if something like a chaperonin protein, which helps to get to the right conformation, is for many, many, many other proteins, for example, to hundreds of proteins, then it seems to me that this argument for that this is just selection for robustness makes more sense, because when it's when you sort of compensate for mutations not in a single counterpart but in hundreds of genes, then the mutational pressure is 100 times more. So yeah.
Yes, but here you make the basic assumption that small gene, low mutation pressure.
Mm-hmm.
On the other hand, it's not the size of the gene, it's what it does. So I could see the argument could work for certain genes that are small gene function that is reduced in the metabolism. Other case, small gene, really important for metabolism. So I would like to see case by case, you know, to see if the argument that you're providing, that would be a counter argument, apply in all these cases.
Mm-hmm.
Depends which gene is redundant. Exactly.
Yeah, but But
at least in that case, you should see it. If the more gene is important in metabolism, the higher the chance to be duplicated. If you don't see that, you would be.
I don't think you see this. Yeah.
Okay. Interesting.
Yeah.
Any question? Yeah.
Thank you, Azat, for the talk, which was very interesting and very informative too. My question actually is probably quite trivial. Do you think that we can interpret the problem of the distinction of neutral evolution versus selection evolution in terms of time scales? That is, can we imagine that there are time scales under which we have to take into account selection, and time scales under which we can admit that evolution is purely neutral, in the sense everything evolves neutrally if the right time scale is considered? And do you think that it is possible in some way to differentiate such time scales, and can this be useful, helpful in some way?
Yeah. It seems that sometimes you maybe can do something like this if you will say that this is precisely what I think sometimes people who talk about evolvability do, and in this example of robustness, robust phenotype allowing you to explore more possible genotypes, so increasing the chance that you will find a very rare but very important adaptation. And if you buy this argument, then you may say that in a very large time scale for this lineage, maybe the fact that something which robustness, which was initially maybe neutral, in a very large time scale, it was beneficial because it made the organism more evolvable and allowed the sort of yeah, I think this is how it may be done, but yeah.
Okay, thank you.
Yeah.
Does this theory CNE apply to the pi in the end or not?
No.
No, okay.
Uh,
So,
I mean-
just here to explain why there is some complexity that are not useless in the end, or?
So why I was talking about I, my idea was to introduce how usually people talk about complexity in evolutionary biology. And the idea was that I is a sort of paradigmatic example because it was mentioned once in the origin of the species, and then it got reprinted from textbook to textbook. And here the idea is that why it was so interesting, because there is something very complex which does a very complex function. So it's not clear how it could have evolved, because it's unexpected that you have a complex system consisting of many interacting parts appearing from nowhere. But if you manage to divide this complex activity into a sort of accumulation of simple activities, then you may see how it evolved by natural selection. But here, the thing is that you see a very complex mechanism which does something which is not that complex. So it's a different kind of problem which is addressed by CNE.
I have a question. Can you use the CNE to illustrate the relation between selection, drift, and developmental constraints? Yeah, you talk about the loss of the CNE is non-selection and is caused by drift and cause a complexity, robust and a complexity structure, which maybe shows evolvability. Yeah, can you illustrate more detail about this relation user sees?
Yes, I think I can. I'm trying to get screen shared again. Okay. There's nothing.
No,
There was something, or
something moved on the screen there,
Okay.
but still we don't see your slides.
and now.
No,
Is this a
it went
picture
back.
of functional complexity?
Yeah, maybe it's.
No, it's unnecessary.
Yes. Okay, I can't manage to.
You can.
But I can just tell. So I had this sort of general conceptual scheme, which had three steps. And remembering you said drift, selection, and developmental constraints, right?
Yeah.
So first there is this A, which is neutral, which is buffers somehow B in regards to possible mutations.
Yeah.
And this buffering already may be seen as a sort of developmental constraint in a very broad sense of development. But if there is some sort of, if it acts in a developmental, during the development. So if A acts on a prior stage of the development, then B appears, right? Or it starts to B appears and continues throughout, then its activity may be seen as sort of developmental constraint on the possible, on how, oh my God, on B.
Yeah.
And then you have just the second step, which is accumulation of neutral or close to neutral mutations by drift, which is again requires some sort of, we should expect that mutations will accumulate. For example, because you have many sites where the mutation can occur, and if you have many sites where the mutation can occur, then indeed we expect that some of them will accumulate, because if you have only one site, it's just such a rare event that maybe it will never occur. But if there are many of them, and many of sites have, many of the mutations are neutral or close to neutral, then indeed they will accumulate. And only after that, we see that in the end, this sort of ratchet, it functions as ratchet because at some moment A is under purifying selection to maintain this initial buffering activity. So sort of all these elements are involved in this process. These developmental constraints may be involved if it's framed in this way, but yeah.
Any question? Yeah, Max.
I don't know how to talk about this, but I'm wondering a bit about the context, obviously the historical context in which the CNE first started. You mentioned this paper by Stoltzfus in '99. So I'm wondering what was the motivation of talking about CNE? So what was the intellectual context where he sort of thought, okay, we need a new way of talking about complexity, and what was its relationship to natural selection? So I'm wondering, what's the relationship in natural selection and CNE in your work, in the work of Stoltzfus, of other people who talk about CNE? Is it just a way of saying, hey, let's think about evolution, not just about selection, about something that is just gives you an adaptation, but let's think about other mechanisms, other ways by which evolution happens. Is this, so to which extent these two mechanisms, if you will, are pitted against each other or are thought to be sort of part of evolution, and sometimes, evolution takes this pathway, another pathway. So I don't know if my question makes sense.
Yeah, so first about historical context. So it's about the example which I didn't present, and I think it will be complicated to present because screen sharing stopped working. So, but he wrote this paper because there was a new phenomenon which was studied, which is an RNA editing in trypanosomal mitochondria. Trypanosomes are the proteins which cause some infectious diseases. And in their mitochondria, there is a very weird thing happening, because after you have your gene, which is transcribed to RNA, and which is normally goes to protein, and we see that in eukaryotes you have an additional step that you have to remove. You have splicing to remove uncoding segments, but in mitochondria of the trypanosomes, you also have an editing. So you also have to insert nucleotides or remove single nucleotides in many sites. And it happens in more than half of the genes. And in some of the genes, you have to insert tens or hundreds of nucleotides or delete some of them just to end up with a functional sequence. And it also seems very weird because it's something which is very rare in all other organisms. And as far as I know, this similar things are known to happen, for example, in humans, but there it's like single event, and usually there is some explanation about very specific protein and why this specific mutation somehow happened this way. But here it's like a mass phenomenon which is just there. And initially, when it was just opened, people were very perplexed about how it could have evolved. And first it was argued that maybe these mitochondria, they accumulated deleterious mutations. And then, because there were some mutations which are deleterious, they evolved sort of this editing mechanism, which will edit their RNAs in order to cancel the effect of these deleterious mutations. But in 1993, there was a group of two biologists who published an argument saying that this actually doesn't work, and it's much more probable that they first had a system which was somehow capable to already correct these errors, and they show that indeed with the enzymes which are already there, it is expected that this kind of activity will be around, maybe not very active, but it will be around. And given that you already have some kind of this activity, you will start accumulating new mutations, and then you will start, because you already accumulated mutations, this activity, it's now essential, so it's under selection. Same thing. So, and was this specific case which was studied, and then Stoltzfus published a more sort of review paper where he claimed that this is actually a specific kind of explanation which is different from what's usually done, because it's in this particular form, and it's maybe used not only to explain this RNA editing, but some other stuff. So this is the context.
Yeah,
And the second is that I don't think that people really like oppose the natural selection and sort of CNE. It's just the case that it seems that, I mean, for me, the concern is that everyone is familiar with how to explain the thing as adaptive. So obviously, when you will try to explain everything as adaptive, you will sometimes succeed, but maybe you will over-explain something which should be explained differently better. And just because maybe CNE as a phenomenon, I think it's much less important overall than simple natural selection. But still, if in some cases it's a better explanation, maybe we should use it. Because maybe it's something which can occur during the course of evolution, so we shouldn't overlook this possibility.
Yeah.
Yeah.
Okay. Yeah.
Yeah, Alexander.
But at the time, they had already genetic drift as other mechanism
Yeah.
of evolution. It's not like they had only selection and
Yes.
it's less revolutionary than you seem to say. They had already.
Yeah, they already had,
Other way.
but the interesting thing is that something like drift is very often associated with increasing variation or deterioration, or it's rarely associated with a novel structure, novel function, or something like this.
Okay. My question was, but unfortunately, you cannot put your slides. Yeah, no, but it's the graph with the mechanical complexity, unnecessary
Yes.
complexity. It went too fast. I didn't understand.
Yes, I can draw it.
Emergent complexity and what is what?
Okay, I can draw it.
So what?
It's actually yes, it's what Friedel.
I still don't understand this graph. Even if it's the second time I see it. And I ask in Namir too, and I still don't understand.
Like these parameters are qualitative or quantitative?
That's a good question.
It would be hard to define a complexity of a
Mechanism, behavior, yeah, that's it.
So, axis. Horizontal axis is the complexity of mechanism. Vertical axis.
Which is defined as number of parts.
I'll go there.
Okay.
And vertical is complexity of behavior. It seems to me that for my purposes, you can keep quite loose with such a definition. But in the paper I took it from, he tried to define complexity as a sort of the shortest description of relevant features of the system. And this was idea inspired by the Kolmogorov complexity of a string. And he thinks that something analogous can be useful in order to analyze the complexity in life sciences and elsewhere. And the idea is that indeed you may separate behavior of a system, sort of activity of a system in a largest possible sense, and sort of the structure of the system, it's the mechanism which is around. And the idea is that if we assume that there is something very simple mechanical relationship between the complexity by adding just new parts or new part types. So here it may be something which has to do with the number of parts and connection between parts and the number of part types, but it's very hard to give precise definition of complexity.
But it's structural. There's nothing about the behavior of the
Yeah, here
system.
it's structural, and here it's about behavior. And the idea is that if everything is simple and mechanical, and as it was with the eye, everything will be on this axis. Maybe it will not evolve always in this direction, maybe sometimes it will go back, but it will be always like this, that as you get more and more complex, happens because you acquire some new functions. And maybe I can lose new functions if you no longer need eyes, or you no longer need something, you may sort of go back, but you will be somewhere on this axis. The second is what is in my work is that sometimes it seems that the complexity of the mechanism increases dramatically while the behavior doesn't seem to be much more complex, which is, for example, in the case of splicing, it may be here there will be a bacterial intron, and here will be a eukaryotic intron plus spliceosome. Because it seems that at the first glance they do the same stuff, they just cut out the intron, but here it was catalyzed by one molecule, and here it's 300 molecules and something, plus spending energy and a lot of steps, etc., etc., etc. And what can be, in fact, maybe it's indeed like that, and this is the answer given by CNE, or something like this answer is given by the idea that it's indeed like that, but the system is more robust. Then there's a question whether we should include this robustness in complexity of behavior or not. It's an open question. I would rather say no, but maybe yes. Or there may be an answer that maybe it seems that it's here, while in fact it's here, if we say that actually every single of these 300 proteins plays an important regulatory role or something like this. That it just seems to be like this, in fact it's like this. And the third axis, which is in Friedelin's paper, what he calls emergent complexity, is to talk about systems which show to be very simply, which appear to be very simply organized, but still do some complex behavior. And this seems to be what people are talking when they're talking about, I don't know, these complex systems theories, when they talk about flocks of birds, which do some very complicated structures, while in fact they're just similar birds which have some sort of similar interactions between them. And in my talk, I was not at all focused on these ones, and I first thought that maybe I should just remove with white this axis, but yeah.
So basically the measure of behavior complexity is always a lower limit, actually.
You mean lower limit?
The fact that, as you were saying, it is possible that there are some interactions that indeed depend from the mechanism complexity, but we cannot trace them back to such complexity. So
here my idea is that to do this kind of, to put the system on this graph, you have to select what in your description is relevant and what is irrelevant. Because if you don't have this criterion, you can describe anything in as complex way as you could. And I think the way to add relevance to the complexity of behavior is to say that relevant is something which contributes to the fitness, to say which is functional. And then you may say that if the only thing which is functional about spliceosome is that it manages to cut out your introns, you will put it on the same level. Maybe if you say, okay, there is some regulation, you will put it there. And then if you add everything, the question is whether you will get to this level or you will still get stuck somewhere in between and you will still see this disbalance. And it seems to me that there is still some sort of disbalance which we observe.
Quick question on the graph. Behavior is used as a function. Because how you do something and what do you do seems to be two different questions. So.
Yes. Yeah, it shouldn't be just a function.
We have a similar point. I don't know if I can follow up.
Yeah, I think you could.
Because I was thinking very similar stuff. Because, for example, for me, the splicing, when you said unnecessary, for me, it's very complex behavior, but nonetheless the very same result.
Right.
Yeah.
Yes.
Function.
Yeah.
So unnecessary is very.
Yeah, okay, I see. It's very normative, that's why I put it here, because I haven't found any, maybe you can call it excessive complexity. I don't
But
know.
you can write a paper to critique this diagram.
Yeah, that's a good idea that,
Because the more you talk about it, the more normal I didn't understand it. I mean, it's garbage. Now, this idea that there's a way to define structure independently of behavior, independently of relevancy, independently of function to make that,
yeah.
the more you talk, the more I think it's not that clear.
Yes, I think the.
How do you define a structure if not in a function? How do you
Yes.
define a behavior
Mostly minority.
but differently than function?
Yeah, that's a good question. I'm thinking maybe if we change behavior on function, then it may be, if we make something like this, if we put here function, and here it will be mechanism plus behavior plus whatsoever, because we cannot really distinguish that. Will it work in this case? I don't know, but maybe. Because then it will work as you said, that spliceosome works in a very complex manner, but with the same function of putting away nutrients.
Yeah, because for me the problem is function, process, result. How can you manage all of this graph? But it's not the complexity of a function is not still here if you are referring to the process or the result of the process. That is not, it's very interesting to reflect on this graph.
But can we distinguish process and the result of the process?
The processes is the unnecessary complex splice
Yes.
protein
Yes.
stuff, and the result is the
Yeah.
splicing, namely the of the neutrons for
But what was elegant in your examples that you present is that it's redundancy, the concept, and that's much clearer.
Yes.
You know, there's redundancy, you could have done the same thing with less.
Yes.
Here you have this intuition, you could have done something with less, but once you think about it, after that you said, yeah, but the relevancy is defined by fitness. Okay, that's already, I have to have an idea of level of selection and where it is and what is the unit of selection.
Yes.
So it's much more complicated, this abstract, than your cases. Your case are, okay, it's redundant. Okay, it could be more redundant. Okay, I can understand. There's too much stuff, too many stuff. But this is very confusing for me.
Yeah, so, but in my case, there's one situation when it's redundancy and it's clear, and there's this other case with when it's some sort of buffering and it's not that clear.
Not that clear.
Yes.
Less
And I
intuitive.
tried to maybe reduce it to redundancy, but I didn't succeed. So that's why I presented as a different type, because, yeah, but maybe you can in some sort of way reformulate it as redundancies, but I can't see.
where does this graph come from?
It's from
Fridolin.
Fridolin's paper, yes, on the complexity. I can post the slides somewhere.
Oh, we saw it in person. We saw him in person present the paper.
Yes, because he came to them. He was my supervisor for master's project, and so the very one day before my master's defense, I thought, oh, this is actually a nice idea. So, I put it inside.
No, but I'm not saying you should not
No.
explore an abstract representation of that
It
kind of
just-
relation.
I
It's
also-
just this graph. The first time you see it, you say, and each time I think about it, no, this makes no sense at all. And that's typical of analogy in philosophy of biology. They run with something, and at the end, nobody understands. But the intuition, it builds an intuition.
Yes.
Because it says to you that there's an equilibrium between structure and behavior, which is not obvious to me. Mechanical is the norm, whatever that means. And if you're there, it's emergent. If you're there, it's unnecessary. So that's the intuition. But on the other hand, I don't know how you do that. Separate structure from behavior. relevancy is fitness and not something else. Because you would like to have a graph like that for toilets, more complex toilets, but it's not fitness. So what would be the relevancy? It would be the function, but
I mean, function, then yeah, then you have a problem whether you can have the same, because some people will claim that it will be the same function in the end, but yeah.
But the function, yeah. But the function is given by us.
Yes.
Fitness is not given by us. That's the elegant. It's the elegance to use fitness as relevancy. You know, the heart as a function not given by us.
No, I agree, but yeah, maybe I'll try to reformulate it with function / relevance to fitness, and then maybe it will work, but I don't know. Yeah, but thank you for.
Any questions? No.
I got other two questions, more general thing. The first one, I would like to push a little bit more on splicing because it's more familiar for me. And for example, there are, seems to be this unnecessary complexity concerning the spliceosome and its process. But I think that also this is for me problematic because now, for example, there are some alternative splicing or mutually exclusive alternative splicing that are very, very complex process that are essential in order to be able to have 1,000 of combination of RNA for a single gene, the DSCAM, etc., the receptor of the.
Yeah.
There are some examples like that. In here, it's very hard for me to say that it's a necessary complexity because it's exactly the complexity that enables us to start from a single gene and to be able to kind of 1,000 possibilities. Not, at the end of the day, the proteins are not 1,000 different proteins. Nonetheless, there is a very big number of these. So this is an idea that I had thinking about is a necessity, and the other. No, sorry. The question is that a necessity is something that you who gave this a necessity to splicing? Is your work or is in the paper some paper? Who said that the splicing process is unnecessary?
I think they didn't.
What you understand is in literature or is
So I'm not sure whether they use the same word, but they say that splicing evolved through the CNE. This is from the very first paper by Sourceforce.
okay.
Yeah, I don't think he uses the word unnecessary, but yeah.
Okay.
I don't know the word, but just the book.
You finished? Or? Yeah. So okay. So first was on about alternative splicing and mutually exclusive splicing and DSCAM and this kind of phenomena. I think it's true that in some cases, phenomena like splicing, they allow a lot of very important complexities, which are very functional. But two things. First, whether it's true in all the cases of splicing which we observe in the current moment. And second, if we have something like an alternative splicing which turned out to be beneficial, it seems to me that it may explain why it is functional now, but I'm not sure that it explains how it evolved, because it seems to me that if you're talking about just alternative splicing as a possibility, it relies on the fact that you just have splicing, and the fact that you just have splicing, it's, I think, would be very unexpected if by just very first introductions of introdes, they will immediately form some sort of interesting, important functional relationships with leading to alternative or whatsoever splicing. So it seems to me that sort of, if the question is why there is splicing at all, then we will say that it may be evolved to a degree neutrally, but then it allowed evolution of many new adaptive traits, such as the examples which you mentioned. Yeah. So in a sense, maybe it can be reframed in this evolvability language, saying that it initially evolved maybe neutral, but it made the system more evolvable, and in the end, it evolved some adaptive innovations, such as.
So it would be an example of your theory of it.
Yes, splicing is used as an example, yeah. Like not every case of splicing now, but why it's evaporated, yes. Yeah.
I did not, I think, clearly understood the second example, the second type of CNE that you talked about the buffering.
Mm-hmm.
Could you go back to it?
Yes, I can do it. I can also draw, yeah.
What is
It really does not work.
No, but I tried to many times. Yeah. Especially, what's the question?
What is not useful? What is not
Yes, okay. Shall I draw? So the idea is that, so the example scenario is the following. There is a protein A and there is a protein B, and it just happened that they physically interact, because it happens that some proteins just physically interact with each other because cells are quite tightly packed, and it's well known that weak interactions may be quite promiscuous, and so they may form sometimes a complex. And the idea is that if they form a complex, then it may be the case that there is a certain mutation in, for example, in B, which is such a mutation that if there is a B without A, which doesn't form a complex with A, but which has this mutation, then in this situation, it's non-functional. For example, maybe on the surface of the protein is some inappropriate amino acid which changes the fold dramatically because it's hydrophobic and it's on the surface, and because there is water all around, then it will ruin everything because the conformation will change. But if there is an interaction, then the fact that there will appear one hydrophobic amino acid, it wouldn't change a lot because it's not in the water outside, it just sticks into another protein. So
And still, the protein is supposed to do its function anyway.
In this case, it's functional.
Okay.
But in the case when it's alone, it's not functional because it changes the conformation. There is also an argument saying that when the complex is formed, the free energy of the complex is lower than the free energy of the two separate proteins, which means that because its free energy is lower, maybe if there was a mutation which will raise the free energy of this functional form, making it less stable, then in the case of the complex, because there is some gain in free energy, maybe we'll tolerate these mutations. This is clear. Yes, and then if we have this kind of mutation, and if there are many mutations which are like that, then we would expect that they will start accumulate by drift, because if there is this complex formation, it's mutual process. So after some generations, it will be not one mutation, which maybe it's not absolutely non-functional, but like reducing function, like 0.9 of initial function. But at some moment, you will have something like this. And then if you have all these mutations and no binding, then it will be absolutely non-functional. And this will mean that now this binding capacity is under strong purifying selection. Because if A loses its capacity to bind with B, then the protein B will be non-functional, hence the thickness of organisms will be much, much lower, so it will be selected against. So now, this situation will be conserved by purifying selection. And what makes it interesting, that maybe it will be not only conserved by purifying selection, but maybe there will be a positive selection for increase of this binding capacity. And this is precisely the moment where you see that it's very hard to put a border between something like C and E and something like a selection for error correction selection, because this scenario will be something like this, the other explanation. The other explanation would be something like there was a functional protein B, then it accumulated a couple of mutations, which made it not non-functional but less functional. And then, because it's less functional, but still okay, but it just happened because mildly deleterious mutations may appear. Then if there is this A, which now can bind, if it was like this and happened like this, now it's beneficial. So maybe A got selected for this activity. And this is the difference between C and E explanation, which says first there was binding, second accumulation of mutations, and the more traditional error correcting or selection for robustness would be something like mutations started accumulating, and then this is why binding happened, because it was beneficial for the organism. Yeah, is it?
But the meaning of neutral, even in the first scenario, is maybe the word is not well chosen. A here allows B to receive new mutation.
Yes.
It's not neutral.
You mean?
It gives B some freedom, some room.
Yeah. So it's neutral not in the sense that it doesn't do anything, but it's neutral in a more population genetic sense that all the processes happen by drift.
Not selected
Not
for.
selected for. This is the meaning of neutral. It's not that it acts. So in this mode of biochemical physiological sense, it's not neutral, but in the evolutionary sense, it's neutral because it wasn't selected for.
And only at the end is it selected.
But it's not, again, it's not selected for, but it's protected by selection. Yeah, so there was no positive selection, but only purifying selection.
Thank you so much, because this is a good scenario. Because it's a similar question. Because I was wondering if it's relevant here concerning purifying selection and the CNA to try to see where is I don't know, maybe it's not relevant, but where is chance, the contingent part, because selection is clear, okay. And here we have the randomness in the accumulation of neutral mutation.
This is for sure.
This is nonetheless strict.
Yes.
Okay. And then there is another randomness that is the occurrence of the binding between A and B.
Yes.
This is another kind of randomness. Okay. No.
Yeah, I agree. And
It's relevant, but I think there are different kind of chance.
yes, it's also to me that these two are different, because, and it also seems that we tried to maybe make some very simple toy population genetic equation to model it, and this accumulation part of it, it's easy. It's very classical. You can estimate this, it's easy because it's just accumulation of neutral mutations. That's it. But this part of how widespread is this binding, but not any binding, but the binding which will allow some neutral mutations. This is kind of questions which it seems that people just don't know the answer. And this is why, at least my attempts to make some sort of toy population genetic models, they all failed, because on this input you have some probability which you don't know at all. And if you make it too small, you say, it never evolves like that. If you make it big, it's always evolves like this. And I haven't found any good estimations. But yeah.
How could you put the probability? Because you need, it's teleologic. So it's about the future. It's a probability that this interaction between the protein would be exactly the one that compensate blah blah blah.
No. It's not like this. The idea is that just how I try to model is that there are two components, and they may mutate, and one of them has the possibility to accumulate these mutations, which will have some deleterious effect if there are no binding, but no or less deleterious effect if there is binding.
Mm-hmm.
And A, it just has sort of two states, it is binding or it is not binding. And I assume that it is binding just means that it acts on, neutralizes all these mutations. And then you can just, because you simplify this to just A is binding, A is not binding, you can just assign the probability of A mutating from one state to another and back. So you can assign these two probabilities, and that's it. So there is nothing teleological in it.
There is an assumption that the two AB have high physical chemical affinities.
If I want to explain the example, yes, but the moment
It's not necessary for them.
yeah, but the moment I do modeling, it all gets hidden into this probability of A binding or not, because I presume that they all count on the correct binding or something like this. And the problem is this, because in fact it's a lot of these physical-chemical assumptions inside, and for any of these assumptions we don't know numbers. In the end, I don't know which numbers to put in the end. Yes. And I'm not clever enough to somehow cleverly solve this model to give the final solution, so I actually had to run it in many generations, and it wasn't really successful. Yes.
Alors, j'ai une question, mais je ne sais pas bien comment la positionner par rapport à ton exposé.
Okay.
Alors, je voudrais savoir si la Constructive Neural Evolution peut s'appliquer au-delà de la génétique.
Oui. Ok, c'est une bonne question parce que en fait, il y a un article de Tyler Brunet qui s'appelle High Level Constructive Neutral Evolution, où il essaie d'appliquer cette théorie à d'autres cas, notamment au cas d'évolution des symbioses, par exemple, pour montrer que peut-être les symbioses, certaines symbioses entre les plantes et les champignons ont formé comme ça parce que peut-être initialement on juste, je sais pas, sécrété différents composants dans le sol ou quelque chose comme ça. Et après, juste parce que s'il y a deux organismes qui synthétisent, synthesize.
Oh, synthesis.
Or synthesize the same compound, then
That's intelligent.
It sounds bizarre.
Okay, which just synthesize the same compound and express it in the environment, then one of them may lose the ability to synthesize this compound because it's just round. And then the other, in case it becomes dependent on this other one, which again is something which creates a dependency between two organisms, but there was never a moment when it was actually beneficial. At least in some cases, because in some cases it may be beneficial to lose, you don't have to synthesize something, you save energy, which is true for microbes, but probably not so true about plants and fungi, because plants and fungi, they are big, and their population numbers are small, and probably something like not synthesizing one component is just very low selection pressure. And also he tried to argue that maybe some organs may have evolved like this, and this, for example, might may have some vestigial organs, because maybe now they are useless, but because we depend on them in some weird ways, just because they were there around, and now we started depending on them for some other things. Even if they don't do anything, we still conserve them.
Okay, and can we say that the CME is a non-selective process?
So there is no positive selection, but there is negative selection. So this is the idea that even negative selection is enough to create this sort of functional relation, if you assume by functional contributing to fitness. And again, in this paper I already mentioned, they explore also this idea how to connect to the exaptation concept. I think the paper is called something like the role of purifying selection in the evolution of functions or something like this.
And when you use the selection, is it only in the sense of the natural selection or another process, another selective process?
Here I talk about natural selection. You can try to apply this kind of thing to cultural evolution. And again, Tyler tried to do this. And I think his case is not really very well informed, because there are immediately questions like selections. What will be the analogy of fitness if you talk about culture? This is what you mean, like cultural evolution, something like this? Or
something like, I don't know, antibody selection? Or neural selection, no?
Okay, but I don't know. I think to like other selection processes, biological selection. Okay, I don't know. I haven't thought about it. I don't think it will work, because in this kind of processes, it's usually something on a physiological time scale, so it's quite short. And here it should evolve, right? So it's evolution in many, many generations. And here you just select once and that's it.
Did we check if Charles or the question from the chat?
Just as he had to go and
Yes, and that's yes.
And other people have to go, and thanks for the talk.
Any question?
Have 4 minutes.
If you another, it's more like no. I was wondering, this is a curiosity, because Stolfus is famous, this is which I know him, concerning the fact that he spent a lot of time trying to prove the fact that mutation is not random, because he wrote a book that was Mountain something, I don't remember the title of the book, and try hard to say that the random mutation are not totally random.
Yeah.
And I was thinking, if this state of mind, it seems that he's against this randomness in biology, had some influence in this other kind of theorization that he provided, and he contributed to the conclusion that you say. This is maybe just it's a, I don't know, it's it's a curiosity that
Yeah, so he's
is a bit biased by this.
by his ideas about mutation biases.
Exactly.
It's a good question because I read 3 articles of Stoltzfus, 1999, 2012, and 2019. 2019, this is his chapter in a book where he talks about, it's called evolutionary causation or something. Yeah, where he talks precisely about all these biases and randomness, and I think he never mentions it in this chapter. And 1999 is the article in which he first calls this a CNE as a sort of general framework to think about many different things in biology. And already there, the way he expresses and formulates it in general terms, he says, like here, I just said, given that they're neutral, they will accumulate. The way he formulates it, he says, when there is this binding, then you have initial state, which was functional without binding, and many other states which are non-functional when there are no binding, but functional when there is binding, which gives that there is a mutational bias from these initial states towards the other ones. So he already formulated some steps of this process in this language of mutational biases. And in 2012 article, he again formulates it in this language of mutational biases, but but I think he didn't incorporate CNE in his more general stuff on biases, and I think it can be done, but I don't know how. Yeah.
Thank you. So, yeah, thank you.